Good morning students, this is 12th class biology, lecture 6 of chapter molecular basis of inheritance. So today we will do the topics transcription in eukaryotes, genetic code and frameshift mutation. So we were talking about the difference of transcription between eukaryotes and prokaryotes, and I told you that in eukaryotes there is requirement of different RNA polymerases for the formation of different types of RNA. So you can say this is a complexity of eukaryotic transcription in comparison of prokaryotic transcription. So this is first complexity in eukaryotic transcription that is found. There is one more complexity, that is the primary transcript. What is primary transcript? That is heterogeneous RNA, means HNRNA. You can say precursor of mRNA. Contains both exon and introns, means after the transcription the mRNA that is formed, that is precursor, okay, that is in premature form, and that is called heterogeneous RNA, and that contain both exon and introns. Now these introns are non-functional and have to be removed. For this it undergoes the following process. So you can say no such process is required in case of prokaryotes. These are called post-transcriptional changes, means removal of these introns. So this is required only in eukaryotic transcription after the transcription. That's why this is a complexity. So for this means you can say introns removal, and for the conversion of heterogeneous RNA into mRNA, it undergoes the following processes. First, splicing. Splicing means introns are removed and exons are spliced, you can say joined together. Then second thing is capping. Here unusual nucleotide, that is methyl guanosine triphosphate, is added to the 5' end of heterogeneous RNA, and this is called capping because here you can say it's like a cap. It is added at the 5' end of the heterogeneous RNA, and what is added? Methyl guanosine triphosphate, right? Then one more change is there, poly-A-tailing, that is also be called polyadenylation, and here adenylate residue, that is AAA, like that, okay. 200 to 300 adenylate residue are added at the 3' end. It is the fully processed heterogeneous RNA, now called mRNA. So after these three processes, means splicing, capping and poly-A-tail, heterogeneous RNA becomes mRNA, and all these three processes come under post-transcriptional changes in eukaryotes. So see here, this is a process of transcription in eukaryotes that is given in your Ncert book. So here you see this is double-stranded DNA, okay, and you have done about the transcription process in prokaryotes, and in the prokaryotes you have seen that RNA polymerase enzyme bind at the promoter region. Likewise here also RNA polymerase enzyme bind at the promoter region to initiate the transcription, but here in case of eukaryotes that RNA polymerase enzyme bind with some transcription factors. So see here, this is RNA polymerase enzyme which is used in case of RNA in case of eukaryotic transcription, okay, and here these many factors are attached here with the RNA polymerase to initiate the transcription. And what are these factors? These are different transcription factors which are required for the transcription process in eukaryotes. But you have done about prokaryotic transcription, and you know that no such factors are required for the transcription in case of prokaryotes. Only a sigma factor was there which was attached with the RNA polymerase enzyme to initiate the transcription, but here these many transcription factors are required, okay. So this is the difference. So whenever you make the diagrams for the eukaryotic transcription, you will show this RNA polymerase along with this transcription factors. These are total 5 transcription factors which are attached with the RNA polymerase to initiate the transcription process. and then the remaining process will remain same. Means first of all there will be initiation with the RNA polymerase and these initiation factors, and one more difference is there. Here this RNA polymerase bind at the promoter region and a specific sequence is present in the promoter region, and what is that sequence? That is TATA, and that is called Tata box. So note down these things. Okay, first you will write the transcription 5 transcription factors are required here, then RNA polymerase at the promoter region having Tata box, that is TATA sequence, right? Then after binding of this RNA polymerase, the remaining process will be like the prokaryotic transcription. It will go ahead in the double strand of the DNA, will open it and will form a mRNA strand by reading the template strand of the DNA, right? All these processes are same. Now one more difference is there. You see here the mRNA which is forming here. This mRNA in case of prokaryote was not having any introns, but here you can see this brown, yellow, then again brown, yellow portion. These are like this. Okay, so these brown segments are presenting introns here. Okay, so these are introns which need to be removed in case of eukaryotes. So these differences you will show in case of the eukaryotic transcription. Otherwise all other things will remain same. Okay, so whenever you describe the eukaryotic transcription, just keep remember these things, and just do it like the remaining things you just do like that prokaryotic transcription, right? Now this is mRNA, new strand of mRNA, or you can say it heterogeneous RNA. At this stage this can be called heterogeneous RNA, or you can say precursor of mRNA, right? Because introns are present here. Okay, now here you can see first of all there are three processes for the post-transcriptional changes in the mRNA, and what are those? Capping, tailing and splicing. So see here, this is capping. Okay, at the 5' end this GPP, okay, this is attached here, so this step is called capping. Then one more step is there, that is called splicing, RNA splicing, where the introns are removed. So how these introns are removed? These structures will be formed. Okay, and these structures are called spliceosome. Okay, and now after formation of these spliceosomes, this will get detached from the RNA, and the remaining portion, means the exon parts, will get attached, and this process is called RNA splicing. Then last one is here you can see poly-A tail, that is polyadenylate residue is added at the 3' end. Okay, so here this green portion is showing AAA like this. It will be polyadenylate residue will be attached here, and this process is called polyadenylation or tailing. Now after all these processes, means after the capping, tailing and splicing, this mature form of RNA is ready, and this is called messenger RNA at this stage. Next topic is genetic code, and genetic code is the sequence of nucleotides that you can say nitrogenous bases in the mRNA, which contains the information for protein synthesis. As you know, by the transcription mRNA is formed, okay, and that has the complementary base pairs that of DNA. Now this mRNA forms the protein. This mRNA has the information for the formation of protein synthesis. But now the difficulty was that how this mRNA can code for protein, means what is the relationship between the amino acids and the mRNA sequence, because up to now you have done about the replication and transcription, and in that in replication DNA is copied from the DNA strand. Okay, DNA is formed on the complementary basis from a parent DNA strand, and in transcription RNA is formed from the DNA strand. So there was a complementarity between the DNA and RNA, but here there is no complementary base pairs are present in the amino acids. Then how the information present on the RNA is used for the formation of that amino acid chain, or you can say for the protein synthesis. So it was a challenge for the scientist, okay, that how can they relate the protein synthesis process for by the mRNA sequence. Now many biologists along with many physicists worked upon it, okay, to know about the relationship between these things, and then they described the genetic code. So first of all, George Gamow, he was a physicist. He said that triplet code is operative as DNA contains only 4 bases while the number of amino acids are 20. As you know, DNA contains only 4 bases that are ATGC, while number of amino acids are 20. Okay, so if one nitrogenous base code for one amino acid, then you can say these bases are not enough to code for the all the amino acids present in human. Then how these can code for the 20 amino acid, how these 4 bases can code for 20 amino acid? Then he said that there can be different combinations, and these combinations can be in set of 3. Okay, so he said that these are present as codons on the mRNA chain in set of 3, okay, to code for the amino acid. So he said that there are total 64 codons that are called triplet codons, because these are present in set of 3, okay, because total 4 nitrogenous bases are present. So if we make the set of 3, then how many combinations will be possible here? If we write the 4 to the power 3 according to the permutation, that is mathematics formula, okay, so if we write 4 to the power 3, then it will come 64. So total 64 codons, 64 triplet codons, you can say, are present here to code for the 20 amino acids. It was first of all, it was told by George Gamow. After that, Hargobind Khorana also tried to give some experimental proof for it, and he synthesized homopolymers and copolymers of RNA molecules. Okay, in laboratory he synthesized RNA molecules. Some were homopolymers, means having the same types of monomer units, and some were copolymers, which were having different types of monomer units with specific combination of bases. Then Marshall Nierenberg also helped in the same process. Okay, he by deciphering the code by making a cell-free system of protein synthesis. So he also made some proteins, cell-free protein, and with the by knowing the sequences, you can say by the codon present in that, he also helped in the same process, means gave the proof that yes, the codons for the amino acids are present in triplet form. For this work, Hargobind Khorana shared Nobel Prize with the Marshall Nierenberg and Holly. That was also a scientist who worked upon the genetic code. Now, based on all these observations, it was finally decided that genetic code is present in the form of triplet codons. So, this checkerboard was prepared, okay, which is showing the triplet codons for the amino acids, for the 20 amino acids present in human. So now we will do the characteristics of the genetic code. So this is a checkerboard showing different genetic codes, and you can see here all these codes are in the form of codons, right? So in the form of a triplet codon, every codon code for a specific protein. Here you can see this UUU, that is coding for the phenylalanine. Then likewise these bases, these codons, you can say, are coding for the leucine. These are also coding for the leucine. Then here these for serine, tyrosine, cysteine, arginine, asparagine. Okay, like this, all these 20 amino acids are coded by these codons. Now how this checkerboard is formed here, for the first position, means for the first letter, this UCAG is written here. Okay, so on the first position you can see here in this row all are having U, 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 U. All the first positions are occupied by U, means uracil. Here for the second position also the same sequence is written UCAG, right? And for the third position here it is written. Okay, here for the each row 4 letters UCAG, 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 means in the same sequence. For the first letter also UCAG, for the second letter also, and for the third letter also. The only difference is here for the each row these all the 4 bases are written here because it's deciding the third position of the base. Okay, right? So now start from here, take the first letter from here, second letter from here, and third letter from here, right? Then it will be U, U, U. Then come to the second, so first letter from here, second letter from here, and third letter from here, so it will be UUC, right? You can see here this is UUC. Then come to this, similarly you take the first letter, that is U, second letter also U, and UU, and third letter from here, that is A. Okay, now take one more example, like here this is first letter, okay, and here this is second letter, so AU, and third letter will be U. Okay, so it will be AUU, right? So like this checkerboard is formed, and in this all the codons for the 20 amino acids are present, and you can count these are total 64 codons are present. So this, you can say, this is the first characteristic of the genetic code. Okay, that genetic code present in the form of triplet codons. Okay, and there are total 64 codons, and out of this 64 codons, 61 code for amino acids, while remaining 3 are stop codons. Here you can see 1, 2 and 3, UAA, UAG and UGA. These 3 are stop codons, and rest 61 are coding for the amino acid. So you can write this characteristic for the genetic code. Next characteristic is codons are always written in continuous manner. No comma, no punctuation held between them. Here you see this is mRNA chain. Okay, and in this mRNA chain these codons are present like this, GUG, CAUs. You can say no comma between these. Okay, and so no punctuation is also present here. That's why we can say this is written in continuous manner. Okay. Next characteristic of genetic code is universal in nature. Universal in nature means if the GUG is coding for valine, then in every organism present in the universe it will code for the valine. Okay, it doesn't happen that in cat GUG is coding for the valine, but in dog it is coding for the alanine. Okay, this is not the case. So the codes are universal. Genetic codons are universal, you can say. GUG always code for valine in every organism. Okay, but there are some exceptions also. Some bacteria are there which are exceptions. Otherwise, it is universal. Like GAG codes for glutamic acid, GCU code for alanine in every organism. See, so it is same for all species. Next characteristic is each codon represent only one amino acid called specific or unambiguous. Okay, here AUG codes for methionine. Okay, it will never code for any other amino acid. So each codon always code for a specific amino acid, and this property of the genetic code is called unambiguous. That's why genetic code is called unambiguous, right? Here AUG always code for methionine, it cannot code for valine. Next characteristic of the genetic code is amino acid which represent by more than one codon called degenerate. Here you can see this valine, okay, and all these codons are different. Here GUU, GUC, GUA and GUG, all these different codons, all these 4 different codons are coding for the same amino acid, and what is that? Valine. Okay, so this property of genetic code is called degeneracy. and that's why genetic code can also be called degenerate, because same amino acid can be coded by different codons. Next property is some codon initiate formation of polypeptide chain, and those are called initiators. Okay, in most of the cases AUG is the initiator codon, and that also code for methionine, that is an amino acid. Okay, so this AUG also code for methionine, and it is also a initiator codon in most of the cases. In some cases GUG is initiator codon, that code for valine. So you can say this is a dual function also for the AUG, that it is initiator codon as well as it code for the methionine. So in most of the cases what will happen in the translation means at the time of protein synthesis, first amino acid will be methionine. So in most proteins, first amino acid that is present is methionine, because the AUG is starting codon also. Now there are some stop codons also, as I have told you earlier that total 64 out of total 64 codons, 61 are coding for the amino acid and 3 are stop codons, okay, which doesn't code for any amino acid, and that's why these are called stop codons, where if these codons come in the mRNA chain, then the process of protein synthesis will stop. So these are called stop codons, and these 3 codons you need to keep remember. These are UAA, UGA and UAG. If these out of these 3, if any one is present in the mRNA chain, then the process of protein synthesis or polypeptide chain synthesis will stop. Next topic we have frameshift mutations. As we have done about the gene mutations in last chapter, and I told you that frameshift mutations we will discuss in next chapter, because here now you have studied about the genetic code. Okay, you know about the codons, then how the mutations will affect the genetic code. So you will better understand about the frameshift mutation now. So you know that point mutation can be due to insertion, deletion or substitution. Okay, substitution means replacement of the base pair. So here point mutation which are due to insertion or deletion, insertion of base pair or deletion of base pair, can cause frameshift mutation later on. See here how these can cause frameshift mutation. Look here, this is a sequence on mRNA having codons for different amino acids. Like here AUG present, which is coding for methionine. Next codon, that is AAG, is coding for lysine. Next codon is UUU, that is coding for phenylalanine. Next is GGC, that is coding for glycine. After that there is UAA, that is a stop codon. Okay, so this is a normal mRNA which is coding for these amino acids and making this normal protein. Right now, due to a point mutation, what can happen? There can be a deletion, insertion or substitution. Okay, so in case of substitution there will be replacement of one nitrogenous base pair. I have already told you, for example sickle cell anemia. Then deletion and insertion type of mutation, how these result into frameshift mutation here. So see here, first case of deletion. Now this was the frame, okay, these were the codons. Now here at this position, like first, second and third, this UUU, this is third codon, right? In this third codon, one U has been deleted, one uracil has been deleted from here. Okay, see here, this uracil has been deleted due to deletion mutation. You can say a type of point mutation. This uracil has been deleted. What will be the impact now due to deletion of this uracil? See here, now what will be the codons now? So first codon will be same as it is AUG, which will code for methionine. Next will be same AAG, that will code for the lysine. But this codon UUU, it was coding for phenylalanine, but now because this U is missing from here, that's why this codon will be like this. It will be read like this, UUG, and this UUG will not code for the phenylalanine. It will code any other amino acid, right? Now all the codons ahead it will be changed, okay, because this has become UUG now. Now the next codon will become GCU, and the next stop codon will change into any other codon, which may be not stop codon now, okay. So this is the impact of the deletion of one base pair. All the frame has been changed, means all the codons ahead this point mutation has been changed now. So such type of mutations are called frameshift mutation, because due to such mutations all the means a whole frame is changing. All the codons present on the mRNA are changing here, and these changing codons will now code for some other amino acids which were forming in normal case. Now those amino acids will not be formed here, right? Some other amino acids will be formed, and a defective type of protein, you can say, will be formed here. So such type of mutations, such type of point mutations result into frameshift mutation. Now see the case of insertion. Insertion mutations also result into frameshift mutation. How? Same thing here also. See here, like this AUG is same as it was in normal case, so it will code for the methionine. Now after it, this uracil has been added here. Before the second codon, that is AAG, one uracil has been added here. Now what will be the next codon after AUG? It will be UAA, and you know UAA is a stop codon, so the translation process will be stopped here, okay. No amino acid after it will be formed. That's why it we can say that it is also changing the whole frame, okay. All the codons have been changed after this mutation. So such type of mutations are also called frameshift mutations. Now see this case. If a whole codon is added, means 3 or multiple of 3 nitrogenous bases are added or deleted, okay. In that case, what can happen? Look here, this first codon was for the methionine, that is AUG, okay. Now a whole set of a whole codon has been added here. You see these 3 nitrogenous bases AAG have been added here, okay. So addition of these bases will not change the whole frame. All the codons ahead it will remain same. Only this additional codon will be there, which will code for a different type of amino acid, right? But it will not change the whole frame present on the amino acid. It will not change the codons present on the amino acids, right? That's why such mutations are not called frameshift mutations, okay, because here 3 nitrogenous bases have been added, and 3 nitrogenous bases can make a codon. So it will make a separate codon. It will not affect other codons present on the mRNA, right? So you can say 3 or multiple of 3 nitrogenous bases if added or deleted, then the mutation will not result into frameshift mutation. Now your homework based on today's topics, you just describe the process of transcription in eukaryotes. Just write it properly, make the diagram as the diagram is given in your NCERT book of the prokaryotic transcription. Just make the diagram like that. Just make the necessary changes that I have told you, okay. And also tell about the post-transcriptional changes within the same question. Post-transcriptional changes, you will tell about the tailing, capping and splicing. Now question number 4 of NCERT I gave as homework in previous lectures, and here the coding strand sequence is given, and you need to tell the sequence of mRNA, okay. So this is sequence in the coding strand, and you know that mRNA sequence is exactly similar to the coding sequence. There are two strands in the transcription unit, or you can say in the DNA, and these two strands are template strands and coding strand. So this is coding strand, okay, and the mRNA that is formed that read the template strand, okay, means that will be complementary to the template strand and will be similar to the coding strands. Just one difference will be there, and what is that? In place of thymine there will be uracil. So see here, this is sequence of mRNA. Here A, in place of T there will be U, then G, C, A, in place of T again there is U, then again GC which is similar to it, then A which is also similar, then in place of T there is U, then GCA, GCA. Okay, so this sequence will remain same, just replace the thymine by uracil. Next question was question number 6 of NCERT, and types of polymerases are asked in the question, nucleic acid polymerases, and there are two types of nucleic acid polymerases that are DNA dependent DNA polymerase, that is required for the replication process, okay. And why it is called DNA dependent DNA polymerase? Because this is depend on the DNA template strand, okay. DNA strand is used as the template strand for the action of this DNA polymerase enzyme. Second enzyme is RNA polymerase enzyme that is required for the transcription process, and this is also DNA dependent RNA polymerase. As you know that in case of transcription also, RNA polymerase enzyme a DNA strand as a template strand. That's why this enzyme is also DNA dependent RNA polymerase. So these two types of nucleic acid polymerases are present, and both are dependent on DNA as the template strand. Next was question number 11 of NCERT, and promoter function is asked here. So promoter is a region of DNA, or you can say it's a part of transcription unit that helps in initiating the process of transcription. It serves as the binding site for RNA polymerase, because RNA polymerase can start the transcription only after binding at promoter. Next was function of tRNA. So tRNA can be called transfer RNA. It's a small RNA that reads the genetic code present on mRNA. Okay, mRNA has the information for the formation of proteins. So what is function of tRNA? It reads the genetic code present on mRNA, and it carries specific amino acid to mRNA, right, on ribosome during the translation of proteins. Now next was exon, function of exon. So what is exon? Exons are coding sequence of DNA in eukaryotes and in prokaryotes also that transcribe for proteins, okay, because we don't use this term in case of prokaryotes because introns are not present there. Whole DNA is the coding whole DNA has the coding sequence, but in case of eukaryotes we have coding as well as non-coding sequence. That's why coding sequences of eukaryotes are called exons that transcribe the proteins.